So, dear students, good morning. So, currently 27 students are watching me live. Everybody, please join YouTube live. I think only 27 people are watching me live. So, what I do, let me share the link on WhatsApp so that everybody can join it and we can start as soon as possible. Sharp 11:40 will start. Okay, bachcho, so I'm back now. Only 49 students are watching me live. So one good news I have to share with you all. As you know, our province was affected from corona virus. Now he recovered and today he joined a department. So province have joined a department now. That's why there is a small get together. So that people were starting, busy in the cutting the cake. So this is the good news I have to share with you all. Due to the prayer of all the students and the faculty members, the professor is now fully healthy and he joined the department now. I don't know whether I'm visible to you guys or not. So 53 are watching me live. Yeah. And the link for today's attendance I have shared in the YouTube comments. So please fill your attendance with the link given in the YouTube. So without much delay, I think already I have wasted 10 minutes. So without much delay, let's start the today's session. Although we have covered all the unit number 1, in unit number 1 we have covered everything. But the few thing is left, that is the unit 2. Now we have started the unit 2. Unit 2, one concept is there, that is the biological file format. But the biological file format, regarding the biological file format, we have already covered, but just only is the small recap. That all the databases, they are representing the biological sequence in their own format. And the way through which we can represent the style in which the databases represent their biological sequences or biological informations is known as simply the biological data file format, biological file format. So this is the biological file format is nothing. This is the representation of data in biological databases. If you guys have any problem or anything you want to say, you please you can comment me. I am watching the comments. Okay. So as I already told you that I have shared the link for both Acme University and for the Villani Science College in the comment box, so you can fill it out to the attendance. Okay, so it's visible. Archana says, Archana, Neha, Pooja, Dipiksha, Shivangi. So currently 61 students are watching me live, and I think the number is more. Why only 60 students? Okay, whatever, let them attend it. So there is a lot of file types we have to discuss. We have already covered the GenBank file format. I think you know already. We have covered in very much detail. Maybe the question, if the exam is of theoretical, then the question may be asked that write down the GenBank file format in detail. So I told you a lot of things was there. The total GenBank file format was divided in 3 parts. One I told you the header, the second one was the feature, and the third one was the sequence. And those are already started. The starting of the GenBank file format is the locus. Then a lot of things was there. We covered in very detail. So there is no need to revise it again. The GenBank file format, you are well aware from the GenBank file format. Then more format, I think you are aware from that the FASTA format. Once you learn about the GenBank file format, GenBank file format is all about the descriptive format, right? In that you get each and everything on which particular date the sequence was submitted, on which particular date the sequence was updated, modified, who were the submitter, from which organization he or she belongs, right? What is the classification of the organism? Where is the promoter? Where is the enhancer? Where is the silencer? If it is being in the written in the annotations. So we covered very detail the GenBank file format. Then I told you that once you are visiting the DDBJ file format, So DDBJ file format is not separate, it is just the copy paste of the GenBank file format. So in the examination, if it is asked to you that you convert what is the DDBJ file format, so simply you have to write on the GenBank file format, because DDBJ people, they directly copy pasted or directly they have adopted the format of DDBJ. I mean GenBank. I think someone is doing the message. Okay. It's not for me actually, but message in different group. Okay. Yeah. So there is no difference between the GenBank file format and the DDBJ file format. So as already discussed, that GenBank file format was a bit kind of the descriptive file format in which you get each and every detail about that particular sequence. But the FASTA format, it's a just type of a very summarized kind of the sequence, file format, in which the FASTA file format starts with the greater than sign in all the cases of the file format. It always starts with the greater than sign, then short description about the sequence, like from which organism it was belongs, what is the SSN number of that particular sequence, right? Very short definition, so that one can recognize that which type of sequence it is, right? And then from the new line, directly the basis starts. It may be nucleotide or it may be protein. So let me make you very clear. FASTA file format is applicable for both, for DNA as well as for the protein. Alright. So it's always start with a greater than sign, short description about the sequence, short description about the sequence, and then from the new line, your ATGC or the amino acid basis starts. Okay. Is it comfortable for everybody? My voice is clear and my PPT is visible to everybody? Please give response. Say yes or no. I'm watching your comments. Thank you. Okay, great. Rajvi, Ruchi, Etvi. Okay. So what I want to say, that doesn't matter which type of sequence it is, you can represent in the FASTA format. Now the question arises, why we need different formats, right? So for that one I told you, once we were discussing about the biological databases, there I told you that since all databases are autonomous in nature, means there is no central administration. Previously there was no central administration. All the organizations are they developed their own databases, so they are controlling them, right? So everybody have their own way for the representation of the data. So that's why this is the reason there is a lot of file formats. And now we bioinformatician or the person who are interested to analyze the data, they may feel bit difficulty to analyze the data. So for example, suppose a very simple problem I have. I want to know the percentage of GC in a particular sequence. Obviously, with pen and paper it is not possible for me to count the number of AT and GC and then go for the calculation of the GC. Alright. So what I can do, I can take the support of the software. So in that particular software, if GenBank file format have been given, and I can download and I can browse that particular sequence in my file format. So I am concerned with only ATGC, alright. But what I am getting in from the GenBank, I am getting each and everything who have submitted, from which organism it belongs to, lot of information. So obviously my software is going to take lot of memory and maybe the calculation it may take bit time. If one or two sequences you have, then no problem. But you have thousands of sequences in a group, then definitely it is going to take a lot of memory, right? So what I can do, rather than go for the description, if I take the summarized form, just start with the greater than sign, short description, and the ATGC. So simply, I can click in the software and I can count the GC percentage. Alright? Thank you. I have taken it. Yeah. It's here. Alright. So FASTA format is better for the analysis point of view. So one more format, like the first multi-FASTA format. Suppose you have 1000 organisms, right? 1000 sequences you have, and you want to save it in your computer. So what you will do? Will you make 1000 file in the FASTA format? No, not at all. What we can open a simple notepad, and in that particular notepad I can put all the FASTA formats, all the files, all the sequences in the FASTA format. Here you can see in this presentation. Very simply you can see, the first it start with the greater than sign, then the new sequence is again start with the greater than sign, then third sequence is again started with the greater than sign. So it doesn't matter that how many sequence I have, I can put on a single file in a single notepad. So that's why it is known as multi-FASTA format. So there is no difference in the FASTA format and multi-FASTA format. The FASTA format means simply you are putting a single sequence in a notepad or in a file. And while you are talking about the multi-FASTA format, in a single notepad you can put many formats. If you are not getting anything, don't worry. Once we go for the practical demonstration, I will teach you how to put multi sequences in a single file. Alright? So don't worry for it. So another format is known as EMBL format. Regarding EMBL format, you are well aware about it. Those who are those who are not able to join the YouTube live, I think this particular thing I am going to keep it open on the YouTube, so later they can join and they can attend. They are not going to lose anything. So regarding the EMBL file format, I discussed a lot. Then the fourth one is known as GCG format. This one we have not discussed. GCG means Genetic Computer Group. They have actually developed the GCG format. In this particular format, there is a space for comment and a space for data, and it is separated by two dots. And it can contain full sequence data like GenBank or EMBL. Means it's also the kind of the descriptive. All the details you can put in this particular format for regarding the particular sequence. Okay. Has a minimum of 6 sequence name length date, means there is the restriction in this particular format. There should be minimum 6 things. Number 1, name should be there, length should be there, type should be there, date should be there, and checksum means how many A, how many T, how many G, how many C is there. So that should be in the GCG format. Alright. See, this is the simple GCG format. The simple GCG format, you can see it here. The checksum is also there. Checksum is how many nucleotide is there, how many bases are there. Type is what? Nucleotide. And here in which particular date it was submitted, and what is the length of this particular length is 774 and 2586 maybe. This is the position of that particular gene in the genome. That may be. Since I have not put it in very detail, that's why I am telling you that here is you can check it out here. That sequence length is 7444 on March 18th, 1999. That was submitted about 10:43. That I have to see what it is. Type N means it's nucleotide, and check means what? Maybe this is the location of a particular gene in whole genome of the organism. And then you can see, just like I have told you in the GenBank file format, there is total 6 columns. So here you can find out 1, 2, 3, 4, 5. So here there is the difference. While you were dealing about the GenBank file format, there was 60 bases in a line, in a row. But in the GCG, there is only 5 columns and 50 bases. Alright. So this is the way you can represent the sequences in the GCG file format. Alright. So this was all about the file formats. I will forward this PPT to you, and you can go through it, and you can do some Google also if you want to know in detail. So now we are coming up with our new chapter. We have discussed a lot what database is all about, why we store the data. Obviously, if you have the data of the biological origin, you are not going to keep it with you. Obviously, you would like to show it to the whole world so that any scientist who is working in this particular area, he don't have to start from the scratch. Suppose I have done the sequencing of a particular gene, and if I keep it secret with me, so the scientist who is working in USA is also working on the same field. So he also have to do the sequencing. That's why we are going to waste the time and effort of the scientific community. So what we can, if any sequencing I have done some annotation. So that sequences, if I can submit in the database, so any scientist working anywhere in the world, on a single click he is going to get the information, so that he can think beyond that, right? And some fruitful result our humanity can get. So suppose up to today's lecture, what we have learned what bioinformatics is all about, right? What is the database, how many type of database is there, how we can retrieve the data from the databases. Alright, so if you make a very good database and it is available to the public, until you don't analyze it, is it going to make any sense? Obviously no. Just take the analogy, just take the example. Suppose if you have a very good library, the thousands of books are there, and if you don't read them, does it going to make any sense? Is there any meaning of having the library if you don't analyze, if you don't read that? Likewise, once you have the database, it is available to all, very good information is there, very good data is there, that may help in the research, but until we don't analyze them, we are not going to get something fruitful. Alright. So now that's why we are now going to read what sequence alignment is all about. What is the sequence alignment? See. So the definition of the sequence alignment is a way of arranging the sequences of DNA. You see the definition? Sequence alignment is a way of arranging the sequences of DNA, RNA or protein. Why we arrange? Why we align? So that we can identify the regions of similarity. हम सीक्वेंसेस को इस तरह से अरेंज करते हैं, चाहे वो डीएनए हो, चाहे वो आरएनए हो, चाहे वो प्रोटीन हो. If you have any kind of a difficulty, please let me know in the chat box. Because each platform has their own pro and cons. The plus point with the YouTube is if you have low internet connectivity and anyhow you have get out of the class, no problem. Later that lecture is in the repository of the YouTube, you can watch it. And in between, once we are taking the lecture from the Cisco WebEx and all, that someone started presenting or some background noise is there, so unnecessarily we are getting the disturbance. So the best part with the YouTube is, even if you have the low internet connectivity, you will be able to attend the lectures, and even if you missed the class, you can watch that total class, whole lectures later on also. So each of the platform have their own limitation, the pro and cons. Although you can the cons is the negative part with the YouTube live is you can't speak up. If you want to ask anything, you have to put your comments in there. You can chat over the comment box. And once I read, I will try to answer you. So sequence alignment में क्या करते हैं, हम इस तरह से sequences को arrange करते हैं, चाहे वो DNA हो, चाहे वो RNA हो बच्चों, और चाहे वो प्रोटीन हो. ऐसा क्यों करते हैं? ताकि उसमें रीजन ऑफ सिमिलरिटी पा पाए कि whatever DNA, RNA or protein I have, multiple DNA sequences, multiple protein sequences or multiple RNA sequences I can recognize the region of similarity कि इनमें सिमिलरिटी कहाँ पे है, कहाँ पर सीक्वेंसेस हैं जो कंजर्व है ना। इवोल्यूशन में इतना इवोल्यूशन होने के बाद में भी इतनी इवोल्यूशन हमारे बीच में हुई है, तो उनमें आपस में बेसेस हैं जो अभी